Hello everyone, I'm Mr. Furlong and you're watching Chemistry for the Biologist. We're going to start our year off talking about chemistry. Seems kind of odd since we're in a biology class, but actually quite a bit of biology is dependent on knowing some things about chemistry. So we're just going to do a very quick review today just on some of the basics of chemistry, things that you would have learned last year in your physical science class. Biology is the study of life, and all living things are made up of matter. Matter is anything that has mass and volume. Matter is typically found in four states: solids, liquids, gases, and plasma. No matter what state it's in, however, all matter is made up of atoms. Atom, we could simply say, is the basic unit of matter. In an atom, there are three subatomic particles: protons, which are positively charged, neutrons, which don't have a charge, and electrons that are negatively charged. In an atom, the protons and the neutrons are in the nucleus. The electrons are flying around on electron shells or energy levels around that nucleus. So as we take a look at this picture, we can see, for instance, that the red circles are representing protons, the yellow ones are representing neutrons, and the blue ones are representing electrons. Now notice we can count the number of protons, and that's going to tell us what element this is. So as we can see, we have 1, 2, 3, 4, 5, 6, 7, 8 protons, which, if we want to know exactly what that is, we can look on the periodic table of the elements. And we find number 8, which has the atomic number of 8, and that is oxygen. So that was one atom of oxygen. In living organisms, we have only a handful of elements. In fact, the carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur will make up 99% of all the elements found in living organisms. So for the most part, that's all the ones that we're going to care about too much this year. Now typically though, these atoms are not found just by themselves. They're hooked up with another element. That makes a compound, two or more different elements combined together. So we might have a lot of hydrogen, we have a lot of oxygen, but we don't find it just as hydrogen and oxygen. It might be combined together to form water, or H2O. Let's take a look at two different types of bonds. The first type is a covalent bond. In a covalent bond, atoms share electrons. Now recall from your physical science class that atoms want to have a complete outer shell. Hydrogen, only having one shell, just needs 2 electrons for it to be complete, but it only has one. So it's looking to share that one electron with another element. Oxygen, having 6 electrons on its outer shell, is looking to gain 2 more. So it will share 2 electrons in order to have a complete outer shell. Here we can see where hydrogen, with its 1 electron, is going to share that with oxygen, which will share one of its electrons. Another hydrogen comes along with its 1 electron and says, hey, I'll share my 1 electron with you if you share an electron with me. Now what we have is a hydrogen that has 1, 2 electrons, and another hydrogen that has 1, 2 electrons in its outer shell. On the other hand, we have oxygen that has 1, 2, 3, 4, 5, 6, 7, 8 electrons in its outer shell, and now it is a very happy atom. And this becomes a very stable molecule. The second type of bond is an ionic bond. Here, atoms will transfer their electrons over. So for instance, sodium has 1 electron in its outer shell. It could either try to gain 7 more or simply lose the 1 that it has, which is what it does. Chlorine, having 7 electrons in its outer shell, is looking to gain 1 more electron. So now we have the case where we have an atom that is looking to lose an electron, another one looking to gain an electron. So this electron in sodium is going to go over to chlorine, Sodium now no longer has that third shell, and it has 1, 2, 3, 4, 5, 6, 7, 8 electrons in its outer shell. Chlorine now has 1, 2, 3, 4, 5, 6, 7, 8 electrons in its outer shell, and it is now a very stable molecule. We take two highly reactive elements, sodium and chlorine, and we make sodium chloride. Now, here's the thing about an ionic bond. If we were to break this bond, say add water to the salt, it's going to separate those two atoms. And what we will then have is an ion. An ion is an electrically charged particle. So sodium, having one more proton than electron, will be positively charged, and chlorine will have one more electron than proton and be negatively charged. In biology, however, we don't usually call them ions. Biologists refer to ions as electrolytes. So in your bottle of Gatorade, you have water and a bunch of ions, essentially. An important ion is hydrogen. We measure hydrogen using something called pH. And specifically, pH measures the concentration of hydrogen ions in solution. As we can see on this pH scale, it runs from 0 to 14. On this pH scale, 7 is neutral. Anything below 7 is considered an acid, and anything above 7 is a base. So the more hydrogen ions we have in solution, the lower the pH is going to be. And as we decrease the hydrogen ion concentration, the pH is going to go up in that basic solution. Now, pH is based on the power of 10. So for instance, when we look at, say for instance, tomato juice, it has a pH of 4, and compare that to orange juice, which has a pH of 3. Orange juice is a stronger acid than tomato juice. Now, because this is based on the power of 10, orange juice is 10 times stronger than tomato juice. Right? But what if we compared tomato juice again with a pH of 4 and lemon juice with a pH of 2? We might think that lemon juice is 20 times stronger than tomato juice, but it's not. Again, this is based on the power of 10. Orange juice is 10 times stronger than tomato juice. Lemon juice is at 10 times stronger of an acid than orange juice. So that means that if we take this being 10 times stronger and this is 10 times stronger, then tomato juice compared to lemon juice is 100 times stronger than the tomato juice in acidity. And if we look at gastric juice, which is the juices found in your stomach, if we compare that to tomato juice with a pH of 4, gastric juice at a pH of 1 is now 10 times 10 times 10, or 1,000 times stronger acid than tomato juice. And the same thing happens with bases. Baking soda has a pH of 9. If we compare that to bleach, it is 1, 2, 3, 4 numbers higher on the pH scale, which means that it is 10,000 times stronger base, the bleach is, than the baking soda. Whether you're talking about a strong acid like a pH of 0 or a strong base like a pH of 14, both are going to be very dangerous. But as a living organism, we need to have a pH somewhere right around 7.4, which is the pH of our blood. And actually, if our pH drops below 7.35, or if it goes above 7.45, that could mean death to us. So we need to make sure that our pH stays relatively the same. In order to do that, we need buffers. A buffer is a chemical that will resist changes in pH, so that when we're eating foods that are very acidic, which most of our foods are. I mean, think about it. We maybe drink some coffee or tomato juice or orange juice. We eat proteins that break down into amino acids. We eat fats that break down into fatty acids. All of those things get into the bloodstream to lower our pH. And so we have to have buffers in place that will resist those changes. Again, we need to have a proper pH if we want to stay alive. So this is what we're going to be taking a look at tomorrow is what type of buffer systems do living organisms have? So I'm looking forward to doing this with you in lab tomorrow, and I will see you then.